the last 1 decade, I am going to present. My laboratory is involved in mostly development of molecular diagnostics for seed-borne pathogens, plant-borne pathogens, food-borne pathogens. Why we are doing this research is because, global population is exploding in the geometric progression, whereas food production is increasing in the arithmetic progression. Population is exploding in the geometric progression. Geometric progression is 1, 2, 4, 16, 64 like that. Whereas food production is increasing in the arithmetic progression in the sense 1, 2, 3, 4 like that. And on the other hand, day by day, arable land is shrinking because of various reasons, because of urbanization, industrialization, and various anthropogenic activities. That is on the other side. On the third part, actually, more than 30 to 35% of the total agricultural produce is going as waste towards pests and diseases. According to FAO data, about 30 to 35% of the total agricultural produce is going towards pests and diseases. And whatever limited amount of arable land with all the difficulties, we need to feed millions and millions of hungry mouths. And again, according to FAO, out of every 5 people on this globe, only 2 people are having balanced diet, that is 2 meals per day. Remaining 3 people are going to bed either hungrily or malnourished. Although we may have produced agricultural commodities more in quantity, but affordability of the people is not there. Particularly if you take Indian context, India is an agricultural country and it is also called as fruits and vegetable basket of the world, because more than 65 to 70% of the India's country's territory is occupied by agriculture and allied sectors, even animal husbandry and agricultural related industries. That is on the background story. On the other side, microorganisms are omnipresent. Microorganisms are present everywhere. More than 90% of the microorganisms are beneficial for human beings, and only 10% of the microorganisms are harmful. Remember, only those 10% of the microorganisms are responsible for causing 35% of the total agricultural loss. And disease resistance is a rule in nature, but disease susceptibility is just an accidental. I told you about 30 to 35% of the total agriculture produce is going as waste at various production stages, starting from the field condition to the warehouse and even after the preparation of food. The total agricultural loss includes even the food commodities. Out of 30 to 35%, there are various groups of microorganisms like fungi, bacteria, and viruses. I have given you what is their share of fungi, their share of bacteria, and their share of viruses. That means fungi are responsible for causing about 15% of the loss, whereas bacteria are responsible for causing 12% of the loss, and viruses are responsible for causing 8 to 9% of the total loss. So when disease can occur, although microorganisms are omnipresent, when diseases can occur, mainly to occur the disease, there are 3 important parameters which should occur concurrently. That means the concurrent occurrence of these 3 important factors leads to the establishment of the disease. So what are those 3 important factors which should occur concurrently? The concurrent factors which are always essential for occurrence of 3 important parameters, those are: pathogenic organisms should be virulent, host organisms should be susceptible, and the atmospheric condition should be congenial. That means a virulent pathogen, susceptible host, and congenial atmospheric conditions are responsible for causing the disease. If any one of these out of these three, if anyone is missing, disease establishment will not be there. That is why all of us, although microorganisms are omnipresent, all of us are healthy because we have got the phenomenon what is known as specificity of the infection or the disease. Although microorganisms are there everywhere, once if you inhale, millions and millions of microorganisms are going inside the body, and once if you exhale, same amount of microorganisms are coming out of the body, but still body is remain resistant because our body has got very, very strong defensive mechanism. At the same time, the specificity of the infection. I told you, disease triangle, the concurrent occurrence of three important factors: virulent pathogen, susceptible host, and congenial atmospheric conditions are essential for establishment of the disease. If any one is missing out of these three, disease establishment will not be there. So I told you in this slide, there are about 15 to 15% of the loss due to fungi, 12% of the loss due to bacteria, and 8% of the loss due to viruses. And mainly, if you diagnose properly, it is easy for any one of us to manage or to control the disease. If the diagnosis is wrong, all other inputs of the disease control strategy is also wrong. That is why the proper diagnostics for this particular disease causing organism is essential. Although these are biotic factors which are responsible for causing the diseases, in addition to these biotic factors, we also have got abiotic factors, atmospheric factors. They are also there to cause the yield loss. But our research is targeted only to diagnose the biotic factors like fungi, bacteria, and viruses, because the proper diagnostics, the proper identification of the disease-causing organism is playing a pivotal role in management practice, in recommending the management strategy. By any chance, if you wrongly diagnose the disease, entire management strategy of the particular disease is going as waste. That is why to control any of these diseases, either at various stages of the crop species, the proper diagnostics, the proper identification of the disease is very, very essential. Many times we wrongly identify, then whatever recommendations recommended for the control, they're all going as waste. So we have also touched upon not only identifying the seed-borne pathogens, the plant pathogens, we are also touched upon something like food-borne pathogens. So food-borne pathogens, what is food? Food is a complex substance derived from either vegetative or animal source, non-vegetative source. Food is a complex substance derived from plant and animal, which is being consumed for nutritional support of an organism. The food safety is monitored routinely by agencies, like various agencies are there to monitor the safety of the quality of the food. Food is contaminated with a number of pathogens, and those pathogens which are transmitted through various routes and which are responsible for causing major health issues, which lead to life-threatening human diseases. Although these diseases are initially occurs at the various levels of the human life, similarly, these diseases which are also responsible for causing the loss in the quality and quantity of the food. Number of foodborne pathogens are reported by World Health Organization and Control Committee for the Plant Disease Control Committee. Among them, Campylobacter, Listeria, Shigella, Salmonella, Staphylococcus. These are some of the important or dominant pathogens which are mostly occurs in the food commodities. So Staphylococcus aureus is one such dominant foodborne pathogen which is colonized approximately about 30% of the human population on this global level and resides predominantly in the anterior nares, harmlessly on the skin, and also in the GI tract of human body, gastrointestinal tract of human body. It causes skin and soft tissue infections, aggressively into bacteremia, and infective endocarditis or sepsis. These are some of the pathogenic symptoms which are there in the human beings. And identification of these pathogenic organisms, there are various conventional stages, conventional strategies to identify these pathogenic microorganisms from the seeds, from the plant, from the food, on all these aspects. And these conventional methods are being replaced by nucleic acid based techniques because of the sensitivity, laborious nature, and complexity to perform. So there are various conventional methods. What are the conventional methods to identify the pathogenic microorganisms in case of plants, or in case of seeds, or in case of human beings or animals? So normally the traditional practice is to first identify the symptomatic portions of the plant body, or symptoms on the human body, or symptoms or the smell, any of these physical appearance. Then carefully identify, carefully isolate the microorganisms from those symptomatic portion of either plant or animal or food, any of these things. Then you need to bring it to laboratory, purify, and subject it for various biochemical tests to identify whether it is fungi, bacteria, viruses. If it is fungi, whether it is aspergillus, it is penicillium, it is Drexlera, it is cladosporium, alternaria, and all these things. If it is bacteria, what are the bacterial organisms which are responsible for causing the disease? And if it is viruses, what are the viral organisms which are responsible for causing these typical symptoms of that particular disease? So identify, then after isolation and purification, you need to subject it for identification by proving Koch postulates. You might have read the Koch postulates. Robert Koch proposed the 4 important postulates. Whether it is human body or animal or plant, the disease-causing organism should be present. This is easy to perform. Disease-causing organism should be isolated and it should be cultured away from the host. And if it is re-inoculated to the host, it should produce similar symptoms which you have isolated the pathogenic microorganism. Those are the Koch postulates. So if you prove Koch postulates, then you will get to know what is the organism which is responsible for causing this particular disease, whether it is fungi, bacteria, or viruses. Then accordingly, you need to recommend the control strategies, fungicides, insecticides, or bactericides, or virucidal compounds, any of these things to control this particular disease. So identification of pathogens in a short period of time is still challenging because it needs a lot of laboratory exercise. So developing easy and sensitive method for identification of pathogens can give more attention about food safety before they are released into the consumption purpose. So the identification, as I said, identification is playing a pivotal role to propose control strategies of this particular foodborne organism. So all these things we have published in this particular journal, which has got very high impact factor. So Koch postulates, I told you, importance of diagnostics, I told you. And today, diagnosticians have an array of methodologies. Traditionally, they are culture-based methods. You need to culture them. You need to purify them. And you need to subject them for various physical, physiological, biochemical methods for identification of these disease-causing organisms. And as they have got laborious practice, nucleic acid-based amplification systems, just like polymerase chain reaction, which is just like a Xerox machine, photocopying machine. If you give the proper primers, any number of copies it can produce, just like a Xerox machine, it will work. When you provide the original and when you provide the cartridge, that is ink, it will give you number of copies. So nucleic acid amplification systems like polymerase chain reaction are promising methods for rapid identification of low number of plant pathogenic fungi or bacteria or viruses. So any of these disease-causing organisms you can identify based on the amplification, nucleic acid amplification systems like polymerase chain reaction. So what are the host and pathosystems which we have used in our studies? These are the host and pathosystems which we have used in our laboratory. Xanthomonas xenopodis or vesicatoria, which is otherwise called as Xanthomonas furfurans in case of tomato, bacterial leaf spot causing pathogen in case of tomato, Xanthomonas oryzae, or oryzae in case of rice, that is bacterial leaf blight causing pathogenic bacteria in case of rice plants, and Xanthomonas campestris, or campestris, which is block rot causing pathogenic bacteria in case of cool vegetables. Cool vegetables like cabbage, broccoli, nal, etc. And the most important pathogen is Ralstonia solanacearum in case of tomato. Although Ralstonia solanacearum is able to cause the disease in over 450 different plants, which belongs to the 50 different botanical families. That means this Ralstonia solanacearum has got very wide host range. It can cause the disease on tomato, on chili, on brinjal, on tobacco, or any of these solanaceous crop species. And as a foodborne pathogen, Staphylococcus aureus, we have tried to identify, or we have tried to develop what is the diagnostic method by which you can identify, or you can identify easily the Staphylococcus aureus. And we have also concentrated on leptospirosis, that is zoonotic disease caused by Leptospira species. These are the host and pathosystems which we have used in our current study to mainly develop proper nucleic acid based diagnostic tests. These are the typical symptoms. Tomato leaf spot, tomato plant showing typical symptoms on the bacterial spot disease, the first picture, and tomato plant showing typical symptoms of bacterial wilt, the wilting of this particular plant. Wilting in the sense it will block this particular Ralstonia solanacearum, blocks the main roads of the plant, that is xylem and the phloem are considered as main roads of the plant by which food and water transportation taking place, and as a result of blockage of that particular main roads, that is xylem and the phloem, the other part of the plant body wilts. So tomato plant showing typical symptoms of bacterial wilt disease. Similarly, tomato fruits, because the marketability of the fruit will be lessened or reduced because of this particular disease, either bacterial spot disease or bacterial wilt disease. So similarly, black rot disease, these are the typical symptoms of black rot on the cabbage or the broccoli or the nalla. The interesting feature here is that, see, unlike fungal pathogens, where fungal pathogens can pierce the cell wall of the plant and get entry into the system, whereas bacterial pathogens can never ever pierce the cell wall, but they always look for natural openings like stomata, hydatodes, gutations, or nectarithodes. So wherever this stomata is there, at the edge of the plant leaf, so in that edge, these bacterial organisms, that is Xanthomonas campestris, or campestris, can accommodate here through natural openings and gets entry into the system, and it will start multiplying in the intercellular system of the leaf. Similarly, bacterial leaf blight causing pathogen in case of the rice plants. See, the distal part of the rice plant showing the typical symptoms, started yellowing, and subsequently it will reaches half of the leaf. Subsequently, the whole leaf will be wilted, or whole leaf will be blighted, so that the photosynthetic area of the leaf will be reduced. Obviously, as a result of the reduced photosynthesis, the yield of the plant will also be reduced. So, because of this particular bacterial leaf blight disease, we can report, or we can find out about 70% of the total loss in the yield is being reported from bacterial leaf blight disease in case of rice. So isolation and characterization of different plant pathogens using biochemical or the molecular methods. So we have collected the seeds from the different agencies, and we have also collected the seeds from the farmers' fields, and we have subjected all those seeds and the plant material collected from the agencies or from the farmers' field for various laboratory screening, either by direct plating or by liquid assay. These are the methods recommended by International Seed Testing Association. ISTA is the authority for recommending the testing of the seeds for bacterial or fungal or any of these pathogenic microorganisms. So after testing, we can subject those isolated pathogens for morphological characterization, biochemical characterization, and also molecular characterization. So these are the colony morphology of Xanthomonas campestris, Petaur campestris on the selective media or on the semi-selective media. Colony morphology, the cream colored colonies on the nutrient agar, that is Xanthomonas xenopodis, Petaur vesicatoria. So this is the typical colony of Ralstonia solanacearum. And now starts with polymerase chain reaction. And we have also standardized various modifications of this polymerase chain reaction. So one such modification is single strand conformation polymorphism. So PCR SSCP, which is most widely used technique for identification of point mutations due to the technique's simplicity, sensitivity, and rapidity. So we have tried to extrapolate or we are trying to use that PCR SSCP for identification of any of these seed-borne bacterial pathogens or fungal pathogens or any other pathogenic microorganisms. So 16S rRNA gene whose use is considered as gold standard for bacterial identification. So this single-strand conformation polymorphism analysis of PCR amplified fragments of 16S rRNA gene has more recently been used as an alternative to genomic sequencing for identification of most of the bacterial species. So we have tried to use PCR SSCP for identification of bacterial pathogens on the seed samples. So the mechanism of this PCR SSCP, after the amplification of PCR products of the target sequence, the amplified product is denatured immediately to two single-stranded DNAs. So the amplified product, when you subject it for heat shock or cold shock, the double-stranded DNA will become single-stranded DNA, ssDNA, and subjected to non-denaturing polyacrylamide gel electrophoresis to identify that single-stranded DNA. Under non-denaturing conditions, single-stranded DNA has the secondary structures. The determination of these secondary structures, these are considered as secondary structures. So these secondary structures, those are single-stranded conformations which are unique to a particular genus, particular species, and also particular subspecies. In case of bacteria, we call the subspecies as pathovars. So under non-denaturing conditions, single-stranded DNA has a secondary structure that is determined by nucleotide sequence. The mobility of the single-stranded DNA depends on the secondary structures of the amplified product. So this formation of loop is unique to the individual species and the subspecies of the organisms. So PCR SSCP, what you need to do is you need to isolate the DNA and amplify with 16S rRNA primers so that you will get PCR SSCP fingerprints, and PCR SSCP fingerprints will be compared with other phytobacterial pathogens for validation. So these are the PCR SSCP fingerprints of various pathogenic species. Those are PCR SSCP banding pattern of Ralstonia solanacearum isolates. We have isolated 150 different strains of Ralstonia solanacearum from different sources. And all those Ralstonia solanacearum is giving unique fingerprinting banding pattern due to PCR SSCP amplification. So this is PCR SSCP banding pattern of Xanthomonas xenopodis with Petaur vesicatoria, the unique banding pattern. So PCR SSCP banding pattern of Xanthomonas oryzae, Petaur oryzae, which is important bacterial leaf blight causing pathogen in case of rice. So these are the individual banding patterns of individual pathogens. But now, if you choose the banding patterns and compare with that of Ralstonia solanacearum at one particular length, Xanthomonas xenopodis at one particular length, Xanthomonas oryzae, E. coli, and negative control along with molecular weight markers. That is how, once if you feed this entire data into the system, then it will only gives you, by comparing these gel pictures, it will gives you more than 99% identity. So what are the applications of this PCR SSCP? So PCR single-stranded conformation polymorphism has simplicity in usage without the requirement of any special equipment, and radioactive labeling is also not necessary. Though DNA sequencing has become more common technique nowadays, the equipment is still unavailable in the common laboratories, and in that way, SSCP has got unique application. So after this PCR SSCP, we have also developed some of the techniques where even SSCP is in need of isolation of DNA, but colony PCR, without isolating the DNA, you can directly subject this pure colony for PCR amplification. And you need not have to isolate the DNA. One step skip you can do, but colony will, if you provide the appropriate primers, it will amplify only when it is a particular bacterial culture's DNA is there. Colony PCR requires isolation of the pathogen, but does not require subculturing, DNA extraction, or even preparation of the sample. Therefore, it's a rapid technique, it's an accurate technique, and it's a cost-effective approach for detection of pathogenic microorganisms. Although we have used all these techniques only with respect to bacterial pathogens, but the same thing you can apply even for fungi, even for other microorganisms. So colony PCR SSCP has an advantage, added advantage because of its rapidity and simplicity. So the procedural details includes very simple. You need to get the bacterial culture. So isolate 6' rRNA primer with amplification, then subject it for PCR SSCP without isolating DNA. You can directly use colony for amplification of the genetic material or the DNA. Colony PCR SSCP fingerprints will be compared with other phytobacterial pathogens for validation. So your DNA fingerprints of that particular pure culture you will get, and by comparing that fingerprints, you can approximately say, oh, this is Xanthomonas, this is Pseudomonas, this is Erwinia, this is Clavibacter, like that. So this is the colony PCR SSCP fingerprints. So we have merged this SSCP along with colony PCR without isolating the DNA of the particular bacterial organisms. Colony PCR SSCP banding pattern of Ralstonia solanacearum isolates. These are the various isolates which we have isolated from different sources. As I said, in our laboratory, we have 150 confirmed Ralstonia solanacearum isolates, and we have been using those isolates for various diagnostic techniques. Colony PCR SSCP banding pattern of Xanthomonas xenopodis, Petavar vesicatoria, that is bacterial leaf spot causing pathogen, that is bacterial wilt causing, and Ralstonia solanacearum is bacterial wilt causing pathogen, that is PCR SSCP banding pattern of Ralstonia solanacearum when compared to Xanthomonas xenopodis, Petavar vesicatoria. So what are the applications with colony PCR SSCP? Colony PCR SSCP, which is being developed in a qualitative tool for pathogen detection and can overcome the isolation of DNA. You can escape the isolation of DNA. Direct colony PCR SSCP for detection of Xanthomonas xenopodis, Petavar vesicatoria, and also Ralstonia solanacearum is relatively rapid and compared to conventional isolation of morphological examination. So now the first technique is PCR SSCP, where DNA isolation step is essential. The second technique is colony PCR SSCP, where DNA isolation step is not essential. So after standardizing this colony PCR and also PCR SSCP, whether we can identify with any of these techniques with multiple pathogenic microorganisms like duplex PCR, when you use two different organisms for amplification, that is duplex PCR. When you are using three organisms for amplification, triplex PCR. Like that, you can also amplify any number of pathogenic microorganisms, different species of fungi, different species of bacteria, along with their host plants, you can identify in the similar gel. That is multiplexing of pathogenic microorganisms. Multiplexing is nothing but amplification of many different pathogenic microorganisms in a same PCR mix without even going for individual isolation of the pathogenic microorganisms. You can amplify both host and pathogen in the same gel, or you can amplify two bacteria, one fungi in same gel, or you can amplify two bacteria, two fungi, two hosts in the same gel. This is called multiplexing. Amplification of many different types of DNA from 2 bacteria, DNA from 2 fungi, DNA from 2 or 3 host organisms. That is multiplexing. Multiplex PCR is a widespread molecular biology technique for amplification of multiple targets in a single PCR experiment. As I said, normally for normal PCR, you will target single PCR experiment. But here, multiple targets you can target. So more than one target sequence can be amplified by using multiple primer pairs in a reaction mixture. So present studies exploits this method for identification of many different pathogenic microorganisms like Xanthomonas campestris, Pithora campestris, which causes the black rot of cabbage, and also Ralstonia solanacearum, which causes bacterial wilt of tomato. So one pathogen from cabbage, one pathogen from tomato, we have tried to amplify in a same PCR mix. So the genes which are chosen for amplification is hypersensitivity response and pathogenicity genes, which direct their ability to elicit HR response. The HRP genes encode a type 3 protein secretion system that appears to be capable of delivering AVR proteins across the walls and plasma membrane of living plant cells. The multiplex PCR fingerprints were obtained, which amplified a 619 base pair of HRP gene from this particular bacteria, Xanthomonas campestris, Pithora campestris, and a 360 base pair section of ITS spacer region from the host. That means here in this experiment, we have tried to amplify both pathogen and the host in a simple single PCR mix. That is, the 619 base pair from Xanthomonas campestris, Pithora campestris, and 360 base pair from Brassica. Brassica is their host for this Xanthomonas campestris. That means both host and pathogen we have amplified in a same PCR mix. So the procedural details includes like this. You need to isolate the black rot causing pathogen, that is Xanthomonas campestris, Pithora campestris, and you need to characterize biochemically whether it is really Xanthomonas campestris, Pithora campestris. Then you need to confirm by molecular techniques with a species-specific primers and also with this HRP gene. So black rot pathogen amplified this 619 base pairs, which is a typical to Xanthomonas campestris, Pithora campestris, and 360 base pair with other host species of Brassica. Similarly, multiplexing we have standardized to isolate and characterize both Ralstonia solanacearum, that is bacterial wilt causing pathogen in case of solanaceous crops, and Xanthomonas oryzae, Pithora oryzae, that is bacterial leaf blight causing pathogen in case of rice. That means the pathogen from tomato and the pathogen from rice mix together and amplify their genetic material by using this multiplex PCR technique. So the standardization of multiplex PCR for both Ralstonia solanacearum and Xanthomonas campestris, Pithora campestris, or Xanthomonas oryzae, Pithora oryzae, you can develop. So in our study, we have developed multiplexing for characterization of Ralstonia solanacearum and Xanthomonas oryzae, two different pathogens. One pathogen is from chili or tomato, the other pathogen is from rice. So these two pathogens we have amplified in a same PCR master mix. So these are the gel pictures of PCR amplification of Xanthomonas oryzae, Pithora oryzae, which compared to 100 base pair ladder, that is, this is the 331 base pair amplified product in case of XOO pathogen, and 281 base pair in case of Ralstonia solanacearum, which is species specific primer. So this test is mainly to confirm whatever microorganisms which we have used for multiplexing are really Xanthomonas oryzae, Pithora oryzae, or Ralstonia solanacearum. So now, when we confirm both Xanthomonas oryzae, Pithora oryzae, and Ralstonia solanacearum by subjecting individually PCR amplification, so when you are mixing both these two for multiplex, this is the picture of multiplex profile, multiplex PCR profile of Ralstonia solanacearum and Xanthomonas oryzae, Pithora oryzae. In both the pathogen in the same gel, you can amplify one at 331 base pair and the other one at 281 base pair. So this indicates one pathogen and this indicates the other pathogen, so that you can reduce the large amount of reagents, which are expensive reagents, by subjecting multiplex PCR. So similarly here, the drawback is the melting temperature you need to standardize very, very carefully, mainly to arrive at melting temperature, which is most suitable for both the pathogen. Both the pathogenic genetic material should amplify at that particular melting temperature. This is the only crucial step for identification or amplification of both these pathogen at the same gel. So here, the annealing temperatures, melting temperature of one is at 58 degrees centigrade and the other is at 64 degrees centigrade, and that is why we have standardized at 61 degrees centigrade. For the normal PCR amplification, one pathogen requires this temperature and the other pathogen requires this temperature. By taking common factor from 58 and 64, we have standardized at 61 degrees centigrade for melting temperature or the annealing temperature of both Ralstonia solanacearum and Xanthomonas variae, Pathor variae, so that you can reduce the consumable cost. So what is the application of multiplexing? Multiplex PCR method plays a crucial role in the identification of several pathogenic microorganisms in the same PCR reaction mixture. The procedure developed in the present studies overcomes laborious, traditional or conventional characterization of phytobacterial pathogens, which we have published in this particular journal. So seed samples can also be directly subjected for multiplexing PCR. You don't need to even isolate the microorganism. Directly you draw the sample, directly you collect the suspected plant material or draw the sample, or even if it is animal pathogen you collect, or even if it is human pathogen, if it is from bacteria you collect, and try to amplify their genetic material so that you can approximately say this is due to this particular microorganism which is responsible for causing this particular disease. Then leptospiral identification. So we have also, leptospira is one of the zoonotic disease. You might be knowing what you mean by zoonosis. Zoonosis is the transmission of the disease from human beings to the domestic animals and vice versa. These are the primers which we have synthesized, which we have designed for identification of leptospira using primer 3 software. And these are the PCR products of leptospira isolates amplified with lipoprotein L. Lipoprotein, which is important protein which is present in the lipid bilayer membrane of this particular organism. So lipoprotein L32 primer, and this is restriction or digestion at lipoprotein L32. So these are PCR products of leptospira isolates amplified with one particular primer, that is LipL41 primer, and RE digestion of LipL41 gene of leptospira isolates. We have also subjected these leptospira for same isolation and amplification. And we have also subjected this leptospira isolate for PCR SSCP analysis. So PCR products were first evaluated for their purity by agrosteel electrophoresis, and this is the procedural details for amplification of PCR products of leptospira, which are mostly amplified at 160 base pair with 16S rRNA primers. So this is the PCR SSCP banding pattern of leptospira species. So this is Leptospira hictioemeraceae, Leptospira javanica, Leptospira australis, Leptospira pyrogenes. These are all different species of Leptospira which you have collected from one of the veterinary institute in Bangalore and subjected those Leptospira isolates for amplification of PCR along with positive and negative control. The positive control and the negative control. You can see one of the unrelated organism. All these leptospira isolates are amplified at one particular wavelength, one particular base pairs, whereas E. coli and the negative control are showing altogether different. So another important method of late which is picking up is audit of MS. So this matrix assisted laser desorption ionization time of flight mass spectrometry can also be used as an effective tool to identify pathogenic microorganisms. See, normally human pathogens which are Staphylococcus aureus, which we have identified using this particular technique, that is audit of MS for MS MALDI-TOF mass spectrometry. So this MALDI-TOF mass spectrometry is an analytical technique in which samples are ionized into charged molecules, and the ratio of their mass to charge can be measured. This is an instrument which measures the analytical technique in which the samples are ionized into a charged molecules, and the ratio of their mass to charge can be measured. By measuring this mass to charge ratio, you can precisely tell whatever the organism which you have used for MALDI-TOF MS analysis is, either fungi or bacteria or virus or nematode or any of these hosts. So in MALDI-TOF mass spectrometry, the ion source is matrix assisted laser desorption ionization. That is MALDI. MALDI is matrix assisted laser desorption ionization. And the mass analyzer is time of flight, TOF. Mass analyzer is time of flight analyzer. This is the equipment. Of late, the importance of this equipment in biology is picking up. So we are depending on this MALDI-TOF equipment for precise, fast identification of microorganisms. Matrix assisted laser desorption, ionization, time of flight, MALDI-TOF, mass spectrometry used to detect and characterize mixture of organic molecules. In microbiology, this MALDI-TOF is used as a rapid, accurate, and cost-effective method for identification of microorganisms. So, normally in the physics, people are using this to identify, or physics or chemistry, people are using this technique to identify and detect and characterize the mixture of organic molecules. Whereas in microbiology or in life sciences, we are using this MALDI-TOF MS as a rapid, accurate, and cost-effective method for identification of microorganisms. Currently, there are two types of MALDI-TOF MS which are currently available or commercially available. That is Vetec MS and MALDI Biotyper. This MALDI Biotyper system, which we have used for identification of Staphylococcus aureus in our research experiments. So mass spectrometry measures mass to charge ratio, MQ, of the ions. Results presented as a mass spectrum, a plot of ion signal as a function of the mass to charge ratio. Spectra used to determine elemental or isotopic signature, the mass of the particles, and the chemical ID or the chemical structure of the particle. The spectra used to determine the elemental or isotopic signature, elemental signature or isotopic signature. Whereas the masses of the particles can also be identified, this mass spectrometry, the chemical identification of that particular particle or the structure of that particular particle, because we will be mostly knowing the cell wall structure, the cell wall identification, the cell wall particles. What is the mass which is present in the cell wall of microorganisms? What is the mass which is present in the cell wall of fungi, cell wall of bacteria, cell wall of viruses? By any chance, if you subject the same cell wall of this biological organism, and this particular equipment will determine, will analyze, and gives us, based on the comparison which is available, already stored data, gives us the appropriate identification of this particular organism. Three components of MS, mass spectrometry: an ion source, the mass analyzer, and the detector. The ion source is a simple, solid, liquid, or gaseous, is ionized. For solids, via electrons or matrix assisted laser desorption ionization. A mass analyzer, ions then separated based on mass to charge ratio, which is done by acceleration and subjecting to an electric or magnetic field. Ions with a lower mass will reach the detector at the faster rate, and the detector records either charge or current produced when ions passes by or hits the surface, produces a mass spectrum that is on mass to charge ratio, usually an electron multiplier. So mold it of an ion source, laser and the ionization chamber, which is there in the picture also, to ionize the sample and transfer into a gaseous space, uses laser energy absorbing matrix to create ions from the large molecules. And this particular analyzer, the mass analyzer, separates ionized analytes according to their mass to charge ratio. And time of flight uses electric field to accelerate ions and measures time to reach this particular detector, which is there here, the detector. So from this ionization chamber, the target plate, all this mass ratio will fuse and reach based on the mass to charge ratio. A detection device is to monitor separated ions. The procedure includes combined isolated colony, that is analyte, with a formic acid and a matrix on moldy plate, followed by solvents to vaporize, leaving only the recrystallized matrix with the analyte. From this culture, you smear on this moldy plate by adding either formic acid and dry and insert into this equipment. Then the moldy biotyper will analyze the chemical component which is present on the surface, and by comparing the existing data in the instrument, it will give us the proper identification of the organisms. So this is the procedural details. So you need to culture, you need to smear on the moldy plate, add formic acid and dry, add to the matrix and insert into this particular instrument moldy biotyper, and it will analyze the data and gives us the proper appropriate identification of the organisms. 99% identity are based on the color, based on the reading. So matrix isolates molecules from each other, protecting them from fragmentation and enabling desorption by laser energy. And matrix consisted of small crystallized acid molecules, usually majority of the cases using synapic acid or alpha matrix or dihydrobutyric acid, then purified water, then organic solvents, alcohol or acetonitrile, then trifluoroacetic acid. So ion source uses UV lasers, nitrogen laser, light wavelength of approximately this 337 nanometers. Then laser pulses fired at the matrix, crystals in the dry droplet spot. Matrix absorbs the laser energy, converting it into an ionized state, and charge is transferred to an analyte. Random collision in the gaseous phase, and ionized analyte and matrix molecules are desorbed from the plate. This is how the target plate is there with a laser beam. These are the molecules of analyte spots, the microbial molecules, along with the matrix spots. When you laser beam hits, the desorption of the molecules taking place, and also desolvation and ionization, followed by proton transfer from this target plate. The proton transferred from the matrix spots to analyte spots and gives us the mass analyzer. It will analyze the mass, charge to mass ratio. Mass analyzer, time of flight. Ionized microbial molecules accelerated through a positively charged electrostatic field into time of flight, that is TOF tube. Inside vacuum tube, ions travel towards an ion detector. The small analytes travel faster when compared to the larger analytes. Ions emerge from the mass analyzer and hit the ion detector, generate a mass spectrum representing the number of ions of a given mass impacting the detector over the time period. This is how the mass analyzer, electrostatic field accelerated, which pulsed UV laser. This is the plate, and separation, time of flight tube. And this detector will detect and gives us mass to charge ratio. This is the matrix and analyte which is subjected for pulsed UV laser, electromagnetic or electrostatic field. This is again how exactly from the microorganisms culture can be smeared and subjected for MALDI-TOF, and it will analyze and gives us the data. Mass spectrum provides profile unique to individual types of microorganisms with peaks specific to genera and the specific to species. Once acquired, compared to a database of a reference spectra, a value in terms of percentage or the score is produced. So foodborne pathogens, what are foodborne pathogens which we have subjected for this MALDI biotyper? All those foodborne pathogens which we have isolated from the vegetarian foods, dairy foods, bakery foods, processed foods, etc. And these are the number of isolates and the representative isolates used for MALDI biotyper. So use of multi-biotyper as a diagnostic tool. Well, so what we have done in our studies is that we have collected the different food samples, subjected for isolation of Staphylococcus aureus, and isolated Staphylococcus aureus was subjected to multi-biotyper either by direct smear or by protein extraction. And also same Staphylococcus aureus, Staph aureus, we have subjected for biochemical characterization and also molecular characterization using extraction of DNA, amplification of PCR, using universal primer, specific primer, and PCR SSCP analysis, along with the comparative study of multi-biotyper. So here our major emphasis is, can we use multi-biotyper for the proper identification of foodborne pathogen? And when we have compared this multi-biotyper data along with our conventional life sciences data, which is exactly correlating with each other, that is why we have concluded we can effectively use multi-biotyper for effective identification of any of the pathogenic microorganisms, whether it is fungi, bacteria, or viruses, or nematodes, or any of these organisms. So these are the different food samples and associated foodborne pathogens. Staphylococcus aureus, Bacillus, Staphylococcus typhimurium, E. coli, Shigella, Vibrio, etc. So these are the colony morphology of different foodborne pathogenic microorganisms. When we have isolated those foodborne pathogenic microorganisms from their semi-selective media, these are the different colored colonies on their semi-selective media. And staphylococcal species associated with different food items. Vegetarian food of so many, Staphylococcus aureus, Staphylococcus epidermis, Staphylococcus hemolyticus. These are the three different species of Staphylococcus species which we have isolated from various food sources: vegetarian food, non-vegetarian food, dairy foods, fruits and vegetables. So foodborne Staphylococcal aureus on selective medium and their biochemical characterization. So we have isolated Staphylococcus aureus and characterized by using biochemical characterization techniques. These are the list of isolates and the sequences deposited on the gene bank using routine PCR amplification techniques. Then comes how exactly we have subjected this confirmed Staphylococcus aureus to MALDI biotyper. So we have used both the samples from the direct smear and also matrix isolated the pathogens and MALDI-TOF MS plate, and we need to insert this plate into the equipment, and this is the mechanism which I have already explained, and the MALDI will analyze the material which is there on this particular culture and it will give us the peaks. And by comparing these peaks, which is already existing data in the data bank, we can say this is the organism, either Staphylococcus aureus or Fusarium or Aspergillus or Clavibacter michiganensis, any organism, any bacteria or any fungi like this you can identify. So how exactly this equipment gives us the data in the form of readings, in the form of color. If this color is, if the range of the reading is from 2.3 to 3, which is highly probable identification of the species in the color, and also it will give the green color and the symbols. So range is from 2 to 2.2, then secure genus identification, probable species identification. This is also colored in green. And if the reading range is 1.7 to 1.9, probable genus identification and not sure of species. And if the range is so much, not reliable. And these are the species, different species of microorganisms, which we have isolated and subjected for multi-biotyper and analyzed based on this particular reading. And this is molded of MS profile of representative protein spectra. Staphylococcus aureus, methicillin susceptible, methicillin resistant, something like that. These are the peak, which is representative peaks of individual organisms. These are the different foodborne pathogens of multi-top MS spectra, either E. coli, Bacillus cereus, Staphylococcus typhimurium, Shigella, Vibrio, etc. Based on this peak, by comparing the data which is already there in the instrument, which will give us the most appropriate identification of different pathogenic microorganisms. So we have compared this data into different strains of staphylococcal strains discriminated by universally conserved thermonuclease gene. So what we have done here is that we have subjected this Staphylococcus aureus for both molecular biology technique and also molded of biotyper, molded of MS biotyper. So both the data are equally comparing with appropriately and confirming the identification of the Staphylococcus aureus. We have also compared this data, molded of MS data, with the PCR SSCP bending pattern, which is confirming the proper identification of moldy biotyper. So we have successfully characterized the foodborne 160 methicillin resistant Staphylococcus aureus species MRSS strains in the present study involving 500 different food samples and showed represented moldy biotyper as an alternative, robust and sensitive tool for the identification of foodborne pathogen, that is Staphylococcus aureus, and we have developed polymerase chain reaction single strand conformation polymorphism, that is PCR SSCP, and also moldy biotyper, and compared. And we have published this data into this particular scientific journal, scientific reports. Then we have also standardized LAMP assay, loop mediated isothermal amplification assay, for proper identification of Staphylococcus aureus. So LAMP assay also we have developed, that is loop mediated isothermal amplification. We have designed the primers and we have subjected for the amplification in the LAMP assay and analysis of the product. The procedural details includes like this. Designed the specific LAMP primers for Staphylococcus aureus, that is SPA gene in, these are the primer designation. These are the primers. And we have amplified the isolated Staphylococcus aureus isolates were confirmed by loop mediated isothermal amplification with the F3, B3 primers. These are the thermal amplification conditions and the components, reaction mixture and the conditions required for amplification of loop mediated isothermal amplification. So bacterial strains used in the assessment of specific LAMP reaction, loop mediated isothermal amplification reactions. So these are the various Staphylococcus aureus, Staphylococcus epidermis, and the other foodborne pathogens like Staphylococcus epidermis, Shigella, E. coli, Bacillus cereus, and Vibrio. All these pathogenic microorganisms which we have used for amplification using LAMP assay. So specificity of the LAMP assay for detection of Staphylococcus aureus. So we have conducted this LAMP assay both in gel assay and also in tube assay. The advantage of this LAMP assay is it need not go for in gel, only at this Eppendorf tube gel in tube assay level you can confirm the isolated pathogen is whether Staphylococcus aureus, Staphylococcus Vibrio or E. coli, any of these things. Specificity of the LAMP assay for the detection of S. aureus. This indicates a gel assay. The specificity of the LAMP assay with the staph or artificially contaminated with E. coli, and the confirmation of LAMP products. The confirmation of the LAMP products with HNB sky blue color for the positive sample on the 2% gel. Sensitivity of the conventional PCR and in-tube assay. So we have confirmed LAMP assay along with conventional PCR. In-tube LAMP reactions on tenfold serial dilution and in-tube assay of LAMP. So we have developed versatile loop-mediated isothermal amplification method for identification of Staphylococcus aureus, and we have published all this data into these various research journals. So I think this is end of my presentation. If you have any questions, you can ask me. Thank you. So these are various techniques which we have used for identification of pathogenic microorganisms, starting from the conventional techniques or traditional techniques, or from the proving of Koch postulates, and up to the advanced technology like multi-biotyper or loop-mediated isothermal amplification. In addition to regular PCR, modified PCR, different versions of PCR like colony PCR, multiplex PCR, SSCP PCR, all these techniques we have adopted in our laboratory for the proper identification of disease-causing organisms. Although we have used one representative organism for identification using all these techniques, but the same techniques you can adopt for identification of any of these pathogenic microorganisms, either which is isolated from human beings, isolated from plants, or isolated from seeds, or isolated from any of the domestic animals, because we have given the representative isolates from seeds, isolates from plants, isolates from the domestic animals, or even the human being pathogen, that is Staphylococcus aureus. Thank you very much.